Ora si registrerà la conferenza.
Okay, still there, I presume. Abcam is currently recognized as a recombinant antibody provider, mainly for research use purposes, which is still our core business, but we are expanding our portfolio by complementary product groups like the multiplexing assays, with the ViaFX products in place, for example, as Katarina mentioned. We have custom antibody developments, conjugation services, added through the latest expedient acquisition, epigenetics, which is mainly based on histone research, and immuno-oncology. Most recently, we added knockout cell lines and gene editing to the portfolio and expanded our expertise through the acquisition of EDGene and ASC. In this webinar, we will focus, of course, on the gene editing segment in particular, the segment which is highlighted with the yellow star. Next slide, please. Okay, the introduction to the cell lines and the CRISPR technique. The initial step in a pharmaceutical research project is usually the usage of regular cell lines or primary cells to test the proof of concept or identify specific mechanisms of action. This is a critical step in the research journey and exactly the place Abcam can bring added value by our gene editing product portfolio. In later stages, the researcher might use animal models or, for example, more specific primary cells that are more physiologically relevant to the researcher journey at that stage. And finally, clinical trials will be set up for safety reasons in a healthy population, and in the latest phases, so in stage 2 and in stage 3, test for efficacy of the target of interest in human patients populations. Next slide, please. Right, the CRISPR-Cas9 gene editing itself. So as I said before, you probably all have experience with the CRISPR technique, but I believe it's relevant to show this slide anyhow. However, the CRISPR-Cas9 is based on a sophisticated defense mechanisms of bacteria against attacks from viruses. The system allows the bacteria to identify and eliminate foreign DNA. In late 2012, or I guess it was beginning of 2013, researchers discovered that the Cas9 enzyme could be reprogrammed to target, eliminate, or even replace specific genetic sequences. This was more or less the birth of gene editing. During this reprogramming, the researcher will design a so-called guide RNA, excuse me. These are small RNA molecules that will match with a specific and no DNA sequences and will bring the Cas9 enzyme to the desired place on the genome where it can do its job. Once on the location of interest, the Cas9 enzyme can silence a gene, so it can induce a knockout by a deletion. It can insert specific sequences, or it can even induce a frame shift that will, of course, lead to the formation of a different peptide in the end. In this case, the phenotype will change. On this slide, you also see a healthcare example of gene editing. Lila, which is a small girl, she was part of twins, and she suffered from leukemia and did not respond to regular chemotherapy. She was treated with engineered immune cells by gene editing, and she recovered from her disease in the end. I will share this presentation afterwards, so you can follow the link that is included here into the lower right corner to read the complete story. I think it's really of interest, and you can see the power of the CRISPR-Cas9 possibilities. Next slide, please. Okay, now I will run through some pitfalls and considerations when using the CRISPR technique. The CRISPR gene editing workflow is very complex, it consists of a great number of stages, and every individual stage might lead to the multiple technical and research considerations during the process. This means that small missteps at any of those stages can result in reprocessing, failure, or even discontinuation of a project. Please keep in mind that the average time a CRISPR experiment will take is approximately 19 weeks, and this is only in an optimal setting. With this in mind, you can imagine that large-scale optimization and automation will increase the potential success rate dramatically and might be even something to consider to outsource. Next slide, please. Okay, the applications. There are many applications for cell lines and lysates. Knockout cell lines are often used in early discovery studies when you want to validate your target and demonstrate that the gene is important for the pathway or the phenotype you are studying. In summary, by using the knockout cell lines, you can perform target validation and identification, antibody validation, and phenotypic assays. You can also validate hits to enhance the efficiency of that hit. The lysates can be used for antibody validation mainly, but they can also be used for signaling pathways, for example, in Western blotting experiments. By using a wider range of CRISPR-generated models, it is even possible to design cell assays for absorption, disposition, metabolisms, and excretion. Next slide, please. Okay. More in general, gene editing knockout cell lines are mainly used as validation tools on one side and as a research tool on the other. As a validation tool, they are very suitable for general antibody validation in, let's say, more or less regular laboratories. But they're also applicable for assay development for antigen detection, for example. On the research side, the applications are more extended. Target identification, disease studies, validation, pathway discovery, and high throughput screening are some practical examples of usage in academic, biotech, pharma, and or with, let's say, within diagnostic companies. At Abcam, we make a distinction between catalog product, meaning on the shelf, and custom. And maybe you can click once more because there's something at the bottom of the slide. Yeah, one more, please. Yeah, there you see the distinguish between the catalog products and the customs. The catalog cell lines are mainly used as validation tools. The custom cell lines are, of course, more dedicated as research tools. Next slide, please. Okay, now I will explain why knockout cell lines are so important for target validation. The slides that we are seeing here represents the so-called 5R framework. It is set up by AstraZeneca and helps to guide their R&D teams in this case, but it's also used in other pharmaceutical companies. And it helps them through the development process in order to improve the phase 3 success rates. Sorry. In essential, you would choose the right target, the right tissue, apply the right safety level, explore the right commercial potential, and identify the right population of patients. By doing this, you mitigate the overall risk of failure and increase the success rate of drug introduction to the market. For this reason, it's important to use physiologically relevant human disease models for validating the right target and confirm that the effect of genes on the phenotype is key to understand the disease and a therapeutic development related to this disease. And I really would encourage you to visit the link. It's a very interesting link. It will show you how much money is lost when you are not applying, let's say, the 5R framework in the correct way. Next slide, please. Okay, so in the previous slides, we have seen that validating the right target is highly important for the pharmaceutical industry, but gene-edited cell reagents can support diverse research areas as well. Because in a research setting, there are in principle 3 areas where these reagents prove extremely valuable, and they are listed here in this slide. Antibody validation by using cell lines or lysates, as discussed earlier. Target selection, for example, using perturbation studies for target expression profiling, and the study of specific mechanisms by using phenotypical assays. In this particular example, you see the change in luciferase activity after astrocyte cell differentiation over time. Next slide, please. Okay. The coming slides will provide some background information on typical Abcam customers, their requests, their challenges, and their needs. The slides are anonymized, but based on real customers. The setting for case study number 1, this is a large biotech company, and we are dealing with a senior research associate who is part of the antibody discovery group. He's mainly focusing on the development of assays for clinical pharmacodynamics. He was not familiar with CRISPR and chose to use Abcam's knockout cell lines and lysates after recommendation by a biological team, as long as the target of interest is available for him. They identified in the end multiple protein targets in a highly expressing human cell background, and were not particular about which cell lines are currently used. Today, they knockout validate their antibodies in immunohistochemistry and flow cytometry as part of assay development for pharmacodynamics and order them straight from our catalog. They need from their supplier, of course, the right target, but also the details of the mutation and the confirmation of the knockout protein secretion, and Abcam delivers them all nowadays. It's also important to highlight here, they see knockouts as the ultimate gold standard because it supports their FDA submissions. Next slide, please. Yeah, thank you. So, the setting for case study number 2. This is an example of a Belgian spin-off from the VIB Research Institute and the University of Leuven. They perform mechanisms of action studies of small molecules. The prospect was a project leader from the startup company, and his focus is on neuromuscular disease therapeutic development, mainly on the so-called Charcot-Marie-Tooth illness. After consultation by Abcam, he decided to buy Abcam's human HGAC6 knockout cell line and the HeLa wild type from the catalog. The right cell line for him was the HeLa, and he needed it together with rest and blood data. The wild type provided as well with limited use licenses. The price was important as well for him, as we are facing here a spin-off company in this case. In the end, he is using the products for the validation of mechanisms of small molecules and uncovered even several biological pathways. The validation and in-depth study of these uncovered targets now provides a first-grade foundation for this customer even. Next slide, please. Case study number 3. Unfortunately, we cannot share the customer details regarding this case, but this is a typical example of a custom project. What we see here are 2 examples of a knock-in construct by gene editing. At case number 1, on the left side, there is an out construct reported by expression rate, as seen left, And above that, confirmed by quantitative real-time PCR amplification, shown in the picture on the right. This is the typical example of a construct for lung cancer research application. At case number 2, we see an astrocyte-specific reporter by incorporation of the GFAP gene by gene editing into a pluripotent stem cell. The reporter is used as a marker to distinguish astrocytes from other glial cells during their during their development. The knock-in cell line expresses an increased luciferase activity over time, as reported in the knock-in genotyping assessment, as well as the functional validation by the luciferase activity. Next slide, please. Okay, this slide has to be read as a kind of a statement. And the statement here is the most widely used method of target validation and to confirm the effect of gene on phenotype is to knock down or to knock out the targeted protein. But we have seen that obtaining highly knockout cell lines and lysates that match your specific experimental requirements can be very challenging for a laboratory, especially if there is a lower level of CRISPR knowledge available, or if there is simply no time to obtain these cell lines at all because of time and funding limitations. The methods that are currently available for acquiring knockout cell lines and lysates are custom services that are currently on the market, but they are recognized as expensive, and it takes a lot of time to have them on your laboratory bench. Commonly offered Hep1 cells tend to be unstable because the rate of mitosis increases, defects in the nuclear spindles form, which results in atypical chromosomes. This is the reason they are not commonly used. Next slide, please. Yeah. Okay. I think it was slide number 16. Yeah, validation is essential to research funding and publication. That's correct. So, when applying for grants and funding at the National Institutes of Health, they can ask you to provide specific information. A few of the requests are highlighted in this slide. You need to assure that you can independently verify the identity and activity of the product before use. When using key antibodies, they might be validated by a recognized technique like Western blotting, ELISA, immunoprecipitation, immunofluorescence, or flow cytometry, using knockdown cells and positive and negative controls, depending on the assay proposed. A knockout cell line or a lysate should be part of your product plan. The recombinant HeLa cell lysate with the EZR knockout is used as a true negative control in this example to validate the product. You see it on the picture on the right side. Next slide, please. Okay, so the question is, why don't scientists validate their experiments? The answer to this question is very simple and straightforward. And I have a cited here, a citat. I don't have the time to do it in the first place, and there might be a lack of knowledge available, as CRISPR is a complex technique. This is what we hear in 70% of the cases, and it's even published in the article you see here on the slide. And they're right. The average time to perform a CRISPR experiment is approximately 19 weeks, so 4 to 5 months in the best case scenario, meaning without performing any repeats at all. Most researchers that are not that experienced will need to repeat their experiments 3 up to 4 times on average before they have the correct edits available. So let's focus on the fact that researchers have a need for knockout cell lines and lysates, but do not have the time nor the funding to obtain them by themselves. So then the question is, next slide, please. Yeah. What if knockout cell lines and lysates are available to order? I think this is the next slide. Yeah, thank you. Okay, so as we have seen, in the best case, it will take around 4 to 5 months from a CRISPR experimental design to a working and stable validation tool. This can be obtained much faster, easier, and with a reliable and stable result by ordering it directly. And there are two options to do this at Abcam. The first option is you pick your cell line or your lysate from the catalog. You will receive it 3 to 4 days later, and you are ready for your target validation experiment. Or, in the second example, you need a tailor-made tool, and you contact Abcam and request a quotation for gene editing services. The Abcam gene editing experts will then closely work together with you and will provide a rapid delivery of cell lines in approximately 8 to 12 weeks, so you can then start with your target validation experiment or with your functional study, depending on your needs. And next slide, please. So, the product overview and the sneak preview. We launched our knockout portfolio in January this year to provide you with high-quality, pre-made knockout cell lines and lysates that are available and easily accessible. They are reliable because they are immortalized mammalian cell lines and are already widely used. They are delivered with relevant genetic and proteomic data. The parental control is available on request, but will be there, of course, all the time. The producing processes are standardized to improve stable reproducibility, and they have the recognized Abcam quality. Above that, a dedicated team of gene editing experts is available on demand and can provide a rapid gene edit product in a time frame around 8 to 12 weeks, as said earlier, depending on your requirements. In general, it will take 8 weeks for a knockout and 12 weeks for a knock-in construct. These gene editing services are available for knock-in, knock-out constructs, but not only for that, also for point mutation constructs. The products and services are very easily accessible by entering the digital catalog on our website. We currently have over 800 knockout cell lines spread over 4 different parental cell lines and about 2,500 knockout cell lysates in 6 different parental cell lines available. And this is just in the catalog. And we have more to come. And I would like to invite Karina to give us a sneak preview regarding the developments in the gene editing area. And maybe, Karina, you can also say something about the COVID developments at the moment.
Yes. So hello,
yeah.
all. Can you hear me well?
Yes.
So my name is Karina Nisa. I'm leading the cell engineering team in Cambridge in Abcam. And if we go to the next slide, I can give you details about what we have in our current portfolio and what we have in the future. So next slide, please. So as Tom said, we have currently 800 knockout cell lines that you can find on our website. Those cell lines have been selected to target multiple research areas, starting from cancer, immuno-oncology, neuroscience, epigenetics, cardiovascular disease, endocrinologies, and, as Tom says, recently infectious disease. And our goal in Abcam is really to serve our customer and the scientists and provide them with the best reagent to really target their research work in a lot of disease area. What we have at the moment is for parental cell lines, so the traditional HEC293s, HeLa, and more oncology cell lines such as F549 and HET116. And because we want really the customer to have more physiologically relevant cells to work with, we are going to work on different backgrounds as well. What we recommend in Abcam is when you use this knockout cell line is to use a corresponding wild type control. So we're providing for each knockout the right corresponding wild type as well. Those knockout cell lines are validated via Sanger sequencing, but also with proteomic. And this is quite important because that's really the final evidence that gives you confidence that the protein is indeed gone from the cell line and from the knockout. So if we go to the next slide. Along the cell line, oh, sorry, the slide before this. Sorry, the slide before. Thank you. So as well as the cell line, we're also providing knockout lysate, and it's about between 2,000 and 3,000 cell lysates, which cover 2,000 genes target, again, in a lot of disease areas, so Alzheimer's, multiple sclerosis, Parkinson's disease, epigenetics, different type of cancers, and immuno-oncology. And we have generated those cell lysates using traditional lysis buffer in different parental cell lines. So again, the F549 colorectal oncology cell line is HET116, HEC293 cells, HeLa cells, hepatocytic cell line like HE2, and MCF7 breast cancer cells. And again, we have the opportunities of providing you with a kit. So we have the wild type lysate that corresponds to the knockout lysate as well. And this is really a great opportunity for any scientist and customer to use those lysates to validate their antibodies, but also to do studies in pathways and see how, when you remove the knockout gene from those cells, what is the phenotypic outcome in the signaling pathway. Those lysates come from cell lines which we have, sorry, I'm still on the previous slide, that have been validated by sequencing data, and also we have validated them by using antibodies against the target. Now the thing that we have covered here is the traditional research areas covered by Abcam, immuno-oncology, epigenetics, and oncology. But recently, we also developed a lot of interest in infectious diseases, and we started by providing a lot of reagents that can help researchers in the fight against COVID-19. So if you go to our website, you can find already antibodies, kits, ELISA kits, Fireplex screening assays to monitor cytokine expression and do phenotypic profiling, but also important cell lines. So we do have cell line that are generated in physiologically relevant background, and what we decided to do is to knock out genes that control the entry of the virus into the cells. So that will enable scientists, for example, to look at the mechanism of entry of the virus, but also develop or understand how a cure or a drug target will be working when some of those important genes are removed from the cells. So that's it. I think we can go to the next slide, and I'm not sure, Tom, if you want to continue the presentation or if you want me to continue it.
No, it's fine. Thank you very much for this clarification on this slide. Maybe I can add something. So if your desired cell line is not available in the catalog, you might simply reach out to us to start up a custom gene editing project to get what you need, but then tailor-made. Okay, so you did a bit of my work, but we are now on the gene editing services, right? Yeah. So summarized, Abcam can offer gene editing services provided by the dedicated team consisting of CRISPR experts. So we have the capabilities to design and deliver knockout, knock-in, and pulp mutation constructs within a time frame between 8 and 12 weeks. And we have access to more than 1,300 cell line models and more than 200 distinct cell lines. The services we offer are customized to your needs and provide low risks because we apply full project consultation, including go and no-go points, mutually agreed between Abcam, and you as a customer. We also deliver fast tracking to your research project by providing investigational new drug and FDA compliant documentation as well. Also, Sigma Aldrich and Broad Institute licenses are available. But please keep in mind that not all competitors can offer all of the required licenses at the moment. At Abcam, we are convinced that our offering of pre-made catalog knockout cell lines and gene editing services helps you to validate your target protein with confidence and interrogate the relationship between the genotype and phenotype, whereby the obtained information is relevant for your observed phenotype in the end. So the next slide, there's a picture in the next slide, I think. Yeah, the picture shows the validation of a knockout of the 125 kilodalton protein of interest with confidence indeed, as you can see here. Okay, next slide, please. So, Abcam's portfolio is already tackling the challenges, not only the challenges related to the Cas9 CRISPR gene editing, but also the issues within other research areas. So we are able to speed up your research and improve the reproducibility of the results. Can provide you a various toolbox for research applications, including knockout validated recombinant rabbit monoclonal antibodies. We have primaries as well as secondaries. We can provide you simple set ELISAs as well as multiplex analysis of microRNA by using of our Fireplex platform. And last but not least, the most recently launched knockout cell lines and lysates, as highlighted in this webinar in January 2020. At Abcam, we are proud that we can offer this novel and powerful technique to our customers, in addition to our expanding product portfolio. Next slide, please. As Karina mentioned as well, I really would like to encourage you to visit our webpage after this webinar and go to the specific cell lines and cell lysate pages. You will find a lot of insights regarding the fascinating world of gene editing and the opportunities it can bring to your organization in general or to yourself as an individual researcher. And I, or Christian, let's see, we will distribute the content of this webinar afterwards, so you will be able to visit all the links that are included in your own environment. Okay, next slide, please. Right. This slide was supposed to show some competitors, but we decided to remove it just before this webinar, so I have to improve a bit on this one. I think the main message is that the current competitors on the market do not have a comprehensive catalog on knockout cell line and cell lines products compared to Abcam. Nowadays, we cannot distinguish a clear market leader for ready-made knockout cell lines and lysates yet, but our aim is to become the market leader for gene editing products and services by CRISPR as the reference knockout technology. As we can offer knockout cell lines as well as knockout lysates validated on genomic and protein level, we are not only capable to deliver true negative controls, but also can supply products usable for the product planning phase, and for example, target selection. Now, on this slide, you see the benefits of Abcam's offering today, let's say more or less in a nutshell. Okay, next slide, please. Good. So with this slide, we reach the end of my story, and I would like to thank you all for your interest and presence at this webinar. Katarina will now open up the questions and answer session to the audience, and I'll